Okay, so here we're going to be learning about DNA and DNA replication. DNA replication. First, let's go through the basics of DNA, what it's made up of, what we should know just to kind of refresh your memory. So, we know that DNA is a double-stranded molecule, and it is known to be semi-conservative, which means that one of the strands is original and the other strand is a new strand. So we know that DNA is made up of 4 nucleotide bases. So we have adenine, guanine, thymine, cytosine. These are just nucleotide bases that are base paired in DNA to specify one's genes and traits when magnified. Dive way deeper into context. You should all know this. I'm just reviewing it, just so it kind of helps for studying for tests. You can just review all this at once. We do know that these are categorized into 2 different names. So we have adenine and guanine. They are known as purines, and thymine and cytosine are known as pyrimidines. They are bonded up between hydrogen bonds. So, but we do know that when these nucleotide bases are paired, we should know that the pairs are between 1 purine and 1 pyrimidine. The 2 base pairs that are added up would be A and T and G and C. So we should know that A and T are bonded together through 2 hydrogen bonds, and G and C are bonded together through 3 hydrogen bonds. When DNA is replicated, enzymes are present to speed up and help the unwinding, replication of DNA and all that. So an enzyme is obviously a catalyst that speeds up reactions. DNA is split up by the enzyme DNA helicase. RNA polymerase builds the DNA, sequences DNA, but polymerase can't sequence and build the DNA without knowing what base pairs are present to complement them. I forgot to mention that DNA strands are built in 5 to 3' direction. The original strand, it's read from 3 to 5, and it's built from 5 to 3. So polymerase builds, but with the help of primase. Primase is the enzyme that lays primer down, lays a bunch of nucleotide bases here so that new strand that will complement them. DNA is in the nucleus. It can't leave the nucleus. How does DNA, genes, traits of someone pass on to every part of the body? Basically, that just is the process of messenger RNA creating a DNA RNA strand to be transferred into the cytoplasm out of the nucleus and send a message to every part of the body. Alright, so this is our DNA. We have DNA in here. Alright, so now mRNA is what's transferred, what's made to leave the nucleus. Basically, it builds an mRNA through transcription, which is messenger RNA, and it's basically a replication so that it can sequence a single strand. The RNA is a single strand DNA. RNA consists of adenine, guanine, cytosine, and uracil instead of thymine and uracil. T is replaced with U, so anyways. So once mRNA is made, this is what it would look like. mRNA has a cap, basically just for protection. And then at the end we have the polypeptide A chain, exons and introns. These are exons, and these ones are introns. So through transcription, the mRNA is spliced, and which removes the introns on the mRNA, and it's now an mRNA with just exons. So here's the cap, so they're all exons. The mRNA can transfer through the nucleus membrane. It's translated into the cytoplasm. tRNA, which means transfer RNA, specific type of DNA polymerase 3 that can synthesize pre-tRNA through transcription. And same process happens. Introns are removed when the tRNA is translated through the membrane, transferred into the cytoplasm once again. Once it's translated, tRNA begins to look T-shaped. It's made up of nucleotides, not amino acids. There's an enzyme. The enzyme is called amino-acyl tRNA synthetase, and every tRNA, like such, attaches an amino acid. After that, same thing happens with rRNA. When rRNA is into one strand, same way, they are spliced into two different subunits. One is a small rRNA, and the other is a bigger rRNA. This occurs in the nucleolus. So they're then transferred into the nucleus. When they're transferred into the nucleus, the two different rRNAs are transferred into subunits and pre-ribosomes. So we have a large, a small. They are then translated out of the nucleus, same way, into cytoplasm. Once in the cytoplasm, the pre-ribosomes, the small and large ones, attach to the mRNA cap. So we have our mRNA. Our top ribosome consists of 3 sites. We have our E site, our P site, and our A site. Then the tRNA attaches to an amino acid. This is what our tRNA looks like. We have anticodons, which is made up of 3 nucleotide bases that match combine to the codons on the mRNA strand. The tRNAs that are attached to the amino acids, like this, inside of a site of the rRNA attached to the mRNA. So tRNAs inside the rRNA attached to the mRNA. Like I said, tRNA has an anticodon, which is 3 nucleotide bases that match up and complement the complementary nucleotide base that are on the mRNA. So once that happens, the ribosome, this one, the rRNA, which is the enzyme, moves along the mRNA strand, and it transfers a tRNA. It moves into the P site, and then once that happens, a new tRNA is moved into the A site, and this one is moved into the E site, and it keeps going basically. And every time it runs out, the tRNA comes out of the E site, basically the exit site, and produces protein, which are the polypeptide chains, and they produce protein. So basically every time tRNA produces another, the amino acids are like coming out of the tRNA, converting into polypeptide chain, into protein, and this is then sent off to the body, and that is what is included in tRNA, mRNA, rRNA, DNA replication.